Hello, ladies and gents. Last week we looked at the organisation in humans, more specifically the circulatory system. So we had a look at the components of blood. We looked at the blood vessels, so how blood is transported around the body. We looked at the structure and the function of the heart, and then lastly we finished off with looking at what happens when the heart goes wrong. And we looked at some specific issues and diseases that a heart can get and how we treat them. This week we're sticking with the idea of an organisation of a species, but specifically we're looking at organisations in plants. Now, remember, plant is a very broad term. So what we're looking at today is the general organisation of plants. And remember that the structures we're talking about today will differ from plant to plant dependent on its typically the location and the environment that it's in. So same format as last week. We're going to start off with a challenge grid. So the first slide has got the questions. The second slide has got a blank challenge grid for you to put the answers. So you can either do that on the PowerPoint or you can write them down. Many people have been sending me in their answers to these mini tasks, and the format in which you do it I'm really not bothered about, but I'm very, very happy to see that lots of you have been sending those in. What I would say is please make sure that you are green penning them, because any answers that you're unable to think of, or more specifically and potentially more importantly, any language that needs adapting, it would be good to see that you do that in either a green pen or, doesn't have to be a green pen. But if there's a way that I can see that you are learning from the feedback, so you go to the answers, which are found at the bottom of this PowerPoint, if you go to the answers, and then I can see you addressing either any answers that you couldn't think of or any answers in which the language could be improved, that would be great. So first of all, your first mini task is that challenge grid. There are a couple of mini tasks scattered throughout this PowerPoint. So we have a 6-mark question again. I'll give you a bit more detail on that when we reach that, but we have a 6-mark question and an opportunity for you to get a little bit creative in producing either a poster or some form of learning resource related to one of the topics that we're going to look at today. So today we're going to look at organisation in plants. There are going to be some similarities in comparison to last week's session, but this is obviously going to be more directed in plants and the structure and function of the plant. So to start with, let's look at the very, very broad organisation of most plants. You will recognise this sort of table/flowchart from our last session, where we looked at organisation in humans. So we follow a very similar pattern when we look at plants. So as always, we start, if we go from the smallest, so at the bottom of that table, we have a cell. And then as we get slightly bigger, we have a collection of specialised cells, and that's what we call a tissue. And then we have an organ, which would be a collection of specialised tissues aimed to carry out a function. And up from that, we have the organ system. And then finally, we're going to see the organism as a whole, which would be the whole plant we're looking at. The example given there is the palisade cell, and the palisade cell is where photosynthesis takes place. So if you want to find the cell which is going to have the most chloroplasts in, that's the cell you're going to be looking for, because chloroplasts are the site of photosynthesis. All the palisade cells are found in a upper layer of the leaf called a palisade mesophyll layer, and that's where all the palisade cells are lined up near the top of the leaf, not the very top, but near the top of the leaf, because that's where they're going to get access to the most sunlight for photosynthesis. Again, looking at the organ that we're talking about here is the leaf. And then going on from that, we have the root system. So the root system stems all the way, obviously, from the roots, and then that carries through to the leaf. The reason why we call it the root system is because if you think about the photosynthesis equation, we require water for photosynthesis to take place. So we call that the root system. Now, on the next slide, I'm not going to have an audio bit on that slide, because I want to talk about that a little bit here as well. On the next slide, all we have is a sort of a open diagram of the leaf. So if you were to cut the leaf in half and look at it through a microscope, this is what you would see. This is the structure of the leaf from top to bottom. So we start with the epidermal layer, and all the epidermal layer is a very thin, transparent, actually waxy layer. And the reason it's waxy is because that helps maintain the water control, because wax is water repellent, so it helps control the water levels in the plant. And the reason it's transparent is because we want sunlight to go through that layer onto the next layer, which is that palisade mesophyll layer, which I was discussing before. So that's where all of our chloroplasts are found, and that's where photosynthesis is going to take place. Moving down from that, we have the spongy mesophyll layer, and it's called spongy for a reason, and that's because if you look at it, you can see lots of air sacs in there, and that means there's lots of ability and opportunity for air to move through that layer. And that allows with our diffusion of carbon dioxide into the plant for photosynthesis and oxygen out of the plant as a result of photosynthesis. So the spongy mesophyll layer is where all of our gas exchange is going to take place. Think of the spongy mesophyll layer as the plant's version of the alveoli in the lungs. Alveoli in the lungs is where oxygen and carbon dioxide diffuse in and out. The spongy mesophyll layer is the same. We also have the xylem and phloem. Now we're going to talk about the xylem and phloem in a lot more detail next, and they are the vascular bundles. So that is where transportation of water, minerals and sugar is going to take place. Now you'll find xylem and phloem scattered all over the plant, and they are effectively the blood vessels of the plant. Now remember, plants don't have blood, they transport different nutrients, but they follow the same function. And lastly, which you don't see in this diagram, but it's important to remember, and we do go in more detail about this in year 10, and we have the meristem. Now the meristem are typically found at the very tips of roots and shoots. So shoots are what we call growing sections of plants, and this is where cell differentiation occurs. So this is where these are effectively your stem cells for your plants. So in adults, our stem cells are found in the bone marrow, and in plants they're found in the meristem. So this is where stem cells are formed, and this is where stem cells become that specialized cell. So that palisade cell that we're talking about here, that would have stemmed from a plant stem cell from a meristem. So if we want to be looking at a specific organ system of a plant, the one that we are going to focus on today is the plant organ system, and that is responsible for the transportation of substances around the plant. So if you look at that diagram on the bottom left of the screen, you'll notice that this includes the stem, the leaves and the roots. If we were to talk about the reproductive system of the plant, we would then be looking at the flowers and the specific anatomy of the flowers and the function with the pollen and the ovum, things like that. We're not going to focus on that so much today. We're going to focus more on the transportation side of things. So that's why we're looking at the roots, stems and leaves. Now, there's a couple of specific things that we can go over here, and we are going to go in a bit more detail, but I just wanted to introduce them a little bit early on. So what we're discussing today is mostly non-aquatic plants. So an aquatic plant is a plant found in water, and plants that survive fully submerged in water. We are not really going to be, or the examples and the learning that I'm going to be talking about today doesn't necessarily apply to them. And that's because what we're talking about is the expulsion of water from the plant, a process called transpiration, movement of water through a plant. And this is heavily controlled by something called the guard cell. So they're found at the bottom of the leaves, and they control the water content that are in the cells. Now, the guard cells are two cells that sit next to each other, and they have the ability to become turgid and flaccid. So when they do this, so when a guard cell becomes flaccid, they close up against each other, as you can see on the right-hand side. So just below this big bulk of text, on the right-hand picture, the guard cells are flaccid, and that means that the stomata, which is the gap between them, as nice and firmly shut. Now, what that enables the plant to do is that enables the plant to retain as much water as possible. So if we think about the weather we've been having recently, not currently today when I'm making this, where we've had lots of rain, but the last couple of days we've had lovely, hot, sunny days. Now, most of the plants in our area during this weather, they would have, during the day, they would have their stomatas closed. So their guard cells would be flaccid and there would be no gaps between those guard cells, which we call the stomata. Now, that means that water can't escape. Now, because the plant wants to retain as much water as possible, and because it's so hot, it's quite easy for the water to evaporate out. Now, if we flip that and we think about the plant wanting to move water through the plant, so let's say the soil is quite saturated with water and mineral ions, and the plant wants to move that water through, then what's it going to do is it's going to make those guard cells turgid. So water is going to flow into the guard cells. They're going to make them turgid, and they're going to open up. And you'll see in that picture there where it says stomata open, that it's then going to produce a hole or a pore in the leaf. And that pore allows gases to exchange, and more importantly, allows water to escape and evaporate off the leaf. So there's a key specific structure in the leaf that we're going to discuss in a bit more detail, and it's important that you know exactly how that works. Now, if we look at the other end of the leaf, at the plant, so we've looked at the leaves with the stomatas and the guard cells. If we now look at the bottom, underneath the soil, at the bottom of the plant, we have the root hair cells. Now, we go on about root hair cells all the time in biology. So when we look at specialised cells, we would have looked at the root hair cell. And you can see from the shape, it has a long protrusion out of the typical rectangular shape that you might be used to. And the reason for this is it increases surface area. And if it increases surface area, then it means that more of the cell surface can be in contact with the soil, which hopefully will have lots of water and mineral ions in it. Now, what that means is that there can be more diffusion of the water, so more osmosis of the water and diffusion and active transport of the mineral ions into the cell. If we did not have that long protrusion from the root hair cell, then all the diffusion and all the movement of substances into the cell would take longer, and it may not be compatible with life. So the stomata control the water levels of the cells, and to get water and mineral ions into the cells, into the plant, we have the root hair cell, which has an increased surface area, which increases rate of diffusion. So now we're going to talk in a bit more specific detail about the transportation of substances through a plant. Now, to do that, we need to become comfortable with the two vascular tissues that are within the plant. Now, we have two types. We have the xylem and we have the phloem. Now, these are the vessels that distribute substances through the cell, and they are all over the plant. So if you look at the diagram here, you can see that in the root, we have not as much xylem and phloem, but they're certainly there to allow transportation of specifically water and minerals. In the stem, so transporting substances up or down the plant, again, we have the xylem and phloem, and in the leaf, you can see the xylem and phloem, and actually in the leaves, they're encapsulated in what we call a vascular bundle. So the xylem and phloem will often run side by side next to each other in what we call a vascular bundle in the leaf. So over the next two slides, we're going to go in a bit more detail about the structure and function of the xylem and the structure and function of the phloem. So the main function of the xylem tissue, this is to transport water and mineral ions only. So we're not talking about sugars yet, we are purely talking about the movement of water and mineral ions through the plant. This will take place in the xylem tissue. Now, something quite special about the xylem tissue is it has a very important function, moving water and mineral ions through the plant. However, it's actually mostly made up of hollow and dead cells that form columns. So on the right hand side of the screen, you can see the sort of diagram of this and how that works. And on the left hand side of the screen, you can actually see a transect image using an electron microscope of what these look like. So the walls of the xylem tissues will be made up of cellulose, because cellulose is what makes up the cell walls of the plant. So even though these are dead, hollow cells, they still have that cell wall mostly made up of cellulose. However, they are strengthened with a secondary substance called lignin. And all this does is it wraps around the cell wall and it just provides a bit more extra strength, because you've got to remember now, we are not metabolizing cells. We are dead cells and they are hollow, so they do have the potential to be quite brittle. So by strengthening them with this lignin, it just provides that extra level of support to ensure that the xylem tissue can carry out their function. Now, if you were to look in a bit more detail, and this is certainly sort of an extension challenge task, if you were to look at the xylem tissue in even more detail, you would see that they're made up of two cell types. We have the vessels, and this is mainly what we're talking about when we talk about xylem tissue, the vessels in which the water and the mineral ions will flow through. And then lastly, we have the tracheids, which is where these are much more narrow cells. And what they actually do is not only allow water and mineral ions to flow up the plant, but they actually allow water to move in a lateral motion, so that's from side to side. And that's very important if you're trying to direct water to a specific area. One of the last key important features of the xylem tissue is that the substances within it, so the water and mineral ions, only move in one direction. Water is drawn into the plant through osmosis, and mineral ions are drawn into the plant by active transport, and this takes place in the roots. And then we want to take those water and mineral ions up the plant, so this only ever occurs in one direction, from the root to the leaf. So only ever one direction for xylem tissue. Now the next form of vascular tissue is the phloem. So we've talked about we've talked about the xylem transporting water and mineral ions, and they are dead, hollow cells, and substances only move in one direction. Now we're going to talk about the phloem. So phloem tissues is where we're going to be transporting our dissolved sugars. So remember, sugars are very important. They are key we produce them in photosynthesis, but remember, plants will also respire, so we need the movement of sugars to take place for respiration to occur. Now, this will take place in the phloem tissue, and the key feature about the phloem tissue is that substances can move in both directions. So, as opposed to the xylem, where water and mineral ions move in one direction, the phloem can move substances in either direction. Now, again, we're looking in a bit more detail here, and we're stretching, challenging ourselves a little bit more, because if you were to dissect the phloem tissue further, you would see that they're actually made up of two main cells. We have the sieve tube cells, and this is mainly where the transport mainly occurs. So these are the tubes in which the sugars will flow through. And then lastly, we have the companion cells. Now, the companion cells have a very, very dense cytoplasm, so they're very packed cytoplasm. And what this mainly does is they actually mainly provide sugars for the sieve tube. And the reason for this is because not only are we transporting dissolved sugars, but we're also transporting amino acids through the phloem tissue. So active transport is often required to take substances out of the phloem tissue where they need to be. So we do need energy for that to take place. And lastly, in the sieve tubes, we have sieve plates. So if you look at the picture on the bottom left, you will see some staggered lines through the sieve tubes, and these are called sieve plates. And all these do, again, is just control that movement. They help maintain the movement of substances through the plant. Without them, you may have movement of sugars happening too quickly and happening in a direction that we're not necessarily happy with. So those sieve tubes just help control that, and as well as that, they do provide some good structure for the sieve tube itself. So phloem tissue, movement of dissolved sugars and amino acids. They're made up of two types of cells, and the substances within the phloem tissue can move in both directions. Here we have a sort of summarization slide of what we've spoken about so far in terms of the xylem and phloem tissue, and it's introducing two key words in which we're going to go into a bit more detail over the next couple of slides. So if we think about the xylem tissue, we now know that the xylem tissue transports water and mineral ions. We know that it goes one way from the roots through the stems to the leaves. And we know that the xylem tissue is mostly made up of dead, hollow tubes that are strengthened with something called lignin. So you can see from the diagram of the tree in the middle there that all the arrows are pointing from the roots, through the stems, up out of the leaves. And that is a process called transpiration. So when we're talking about transpiration, we're talking about the movement of water through a plant. So now if we go to the right-hand side of the page, we look at the phloem tissue, which we know transports dissolved sugars, and those sugars are made as a result of photosynthesis in the leaves, and they will be transported typically from the leaves down into storage in the roots, but this can be in any direction. Now this process is called translocation. So translocation is the movement of sugars through a leaf, sugars through a plant. Transpiration is the movement of water. We're going to dive into those two things in a bit more detail now. So here we're going to really consolidate the idea of translocation, so the movement of sugars through the plant. So I think the diagram on the right really nicely illustrates what we're trying to talk about here. So I've already mentioned that translocation takes place in the phloem, and it's the movement of dissolved solutes, so that's going to be sugars and amino acids. Now what we are going to do is introduce two new terms now, because when we talk about translocation, we talk typically about it occurring from a source to a sink. Now, sink is a bit of an odd word, but it does make sense if you bear with me. And source does make sense when you think about this is where the sugars are sourced from. So if I tell you that, where are the sugars sourced from in the leaves, we should think and we should be able to know that, okay, well, where do sugars come from in the plants? So that takes place in the leaf because that's where photosynthesis occurs. So the source, when we talk about translocation, is always the leaves. It's always where photosynthesis is taking place. So you can see from the diagram that we have a leaf cell and we have some chlorophyll in that cell, and we can see that sugars, in this case, a form of sucrose, are being formed. So what's going to happen here is those sugars are going to be moved into the companion cell, and then from that they're going to be inserted into the phloem. And from the phloem they're going to flow through the plant, and typically, remember, not always, remember they do move in both directions, but typically in a plant, most of the sugars are going to head down to a sink. Now a sink is where they are stored. So in most cases in a plant, this is in a root cell, and they will travel through the phloem, again through a companion cell into the sink, which is typically a root cell, which is where plants store their sugars for if they need them, for example, in an evening or overnight when there's no access to sun. If plants need some sugar for respiration, they can access this through their sink. Or if we're looking at the winter time when we have less light and less access for plants to photosynthesize, and they're going to need a bit more sugar, they can always go back to their reserve storage of sugar, which is going to be found in their sinks, in this case a root cell. So translocation, movement of sugars, both directions, from a source, aka a leaf, to a sink, aka a root cell. So now transpiration. So transpiration, as we know, is the movement of water and mineral ions through a plant. This is going to take place in one direction. It's going to take place from the roots up through the stem to the leaves. And with this, we have something called a transpiration a transpiration stream. So a transpiration stream is a constant flow of water through the plant. So if you think of a stream as a trickling of water constantly, we can think of that same in the plant. Now, this is all powered by the evaporation of water from the leaves. And this is something called water potential, which actually we go into in a bit more detail at A-level biology, but it's always worth introducing the idea now. You don't need to know the specifics of water potential, but what we do need to know is as water is evaporated from the leaf, we introduce something called a negative pressure. Now, what that means is you can think of it almost like a vacuum up the top of the plant. And what that's going to do is as water evaporates off the leaf into the atmosphere, that leaf is going to want to bring more water from below up back into the leaf. So it's going to pull that water from the stem. And then the stem is again going to want to be drawing more water, so it's going to draw it from the roots. And then the root again is going to want to draw more water, so it's going to get that from the soil. And this produces a constant transpiration stream. And remember, that all started from simply the water evaporating from the leaf. So because of that negative pressure in the leaf, it means that we are constantly moving water and mineral ions through the plant from the roots all the way to the leaves. Now, we can go in a little bit more detail when we look at transpiration, and I did speak about this earlier, but we're going to look at the idea of the stomata and the guard cells in a bit more detail here. So remember I said the stomata are small pores that are found in the leaves, and they have the ability to open and close dependent on the water levels of the plant. So if you look at the diagram here on the left-hand side, you can see this very nicely. And notice it does talk about solute potential. And again, remember we're sort of dipping our toes into A-level here, but I'm going to try to explain it in a way that's accessible now. So if we look at the diagram on the bottom left, the left-hand stomata, or stoma with the guard cells, you can see that water is going into the guard cells. So what this means is that there is excess water in the plant. If there is enough water to be going into the guard cells via osmosis, that means the plant has enough or too much water. So that means it wants to get rid of some. So what it's going to do is it's going to move that water into the guard cell, which is those two green curvy cells you can see there. And what that water is going to do is it's going to flow into the vacuole of the cell. And then what that does is it makes the guard cell turgid, and it opens up the stomata. So then now we have an open pore in the leaf, and then any water from the leaf can now evaporate out of the stomata into the atmosphere, thus getting rid of some of that excess water. Now, if you look at this on the opposite side, if the plant is lacking water, then you can see on the right-hand side of that diagram, water is leaving the guard cell because it needs to retain it elsewhere because it's lacking water. So then what it's going to do is the water is going to leave the vacuole, the guard cell is going to become flaccid, and it's then going to close that stoma. So, sorry, just to clarify, stoma is the singular term, stomata is plural. So if we're talking about lots of pores in the leaves, we're talking about the stomata. If I'm talking about one specifically, it's the stoma. So if we can think about this process of how water is controlled in the leaves, what we want to be talking about here is the idea of water going into the guard cell, which makes the guard cell turgid and opens up that stoma. And conversely, if the plant wants to retain water, water is going to leave the guard cell. That guard cell is going to become flaccid, and thus the stoma is going to close. Now we have your next mini task, and I know you're probably thrilled looking at this slide, but I would like you to have a go at a 6-mark question. So this question shows us a diagram of the direction of movement of substances through a plant, and it's asking you to describe how ions, water, and sugar are obtained and transported through plants. So if you go back over the last couple of slides, you will know that when we're talking about the transportation of ions and water, we're talking about transpiration. This occurs in the xylem. And if we're talking about sugars, we're talking about the transportation through the phloem. So if you don't want to write the full answer for this, I would at least encourage you to make some notes of how you would answer this. Because although this is a 6-mark question, this is one of the examples of a 6-mark question that you could actually break down into 2 3-mark questions. So split it in half. So I want 3 marks on how ions and water is transported through a plant, and then 3 marks on how sugars are transported through a plant. If you think of it that way, it might be a little bit less daunting than thinking of this as a 6-mark question. 2 paragraphs, 1 on water and ions, 1 on sugars. So the last couple of things we have to talk about in terms of the organisation of the plant is we are sticking with the idea of transpiration. So we're focusing a bit more on the idea of transpiration and the movement of water than we are translocation and the movement of sugars. And that's because we have to learn how or what factors can affect the rate of transpiration. So what factors can affect how quickly water moves through a plant? So the 4 factors we have to be aware of are temperature, light intensity, humidity, and air movement. So for the top marks, you want to be, first of all, being able to identify these 4 things. So you have to remember that these 4 are the factors. To get a few more marks, you want to be telling me how they affect the rate. So do they increase the rate? Do they slow the rate? And then for the absolute top marks, you want to be explaining that. So how exactly does temperature increase the rate? And how exactly does humidity reduce the rate? So let's talk about those in a bit more detail. So I've included the graph for each of these. So I've got the factors on the x-axis and then the rate of transpiration on the y-axis. Now you can see that temperature and light intensity follow a very similar trend. So temperature and light intensity will both increase the rate of transpiration as you increase that factor. So if we stick with temperature here, so as you increase temperature, you're going to increase the rate of transpiration. Now the reason for that, to explain that and why that happens, is because if you increase temperature, you're going to increase the kinetic energy of the individual water molecules. Now if they move faster, that means they're going to find it more easy to evaporate off the leaf. And again, thinking back to the transpiration stream, as water evaporates off the leaf, you're then going to be drawing more water through the stem and through the roots. However, if we reach very high temperatures, then the stomatas will close because what the plant doesn't want to do is lose too much water. So as temperature increases, so does the rate of transpiration. However, there is a limit because the plant doesn't want to lose too much water, so it will close those stomatas, close the stomata, thus preventing any more water loss. Light intensity is the same. So light intensity, you're going to increase the rate of transpiration, and that's because as you increase light, you stimulate stomata to open because photosynthesis is increasing, and we want water for photosynthesis to take place. So as the light intensity increases, so does the rate of transpiration to encourage more water to flow through the plant. However, if we have a high light intensity, what that typically brings with it in the natural environment is high temperature. So this is where we see the same trend as the temperature. So if we have a too much or if we have too high a light intensity, that's going to heat the leaf, and again it could lead to excess water loss. So it's going to close the stomata and slow down the rate of transpiration. Now, humidity. So humidity is the concentration of water in the air, in the atmosphere. Now, as humidity increases, transpiration actually decreases. So as you have more water in the atmosphere around the plant, the rate of transpiration is going to decrease. And the reason for that is because if we think about the rules of diffusion, so diffusion is the movement of substances in high concentration to low concentration. This means that if there is not a lot of water in the atmosphere, that means that water is going to readily evaporate off the leaf because there's not a lot of water in the atmosphere. So it's going to want to go to that area of low concentration. However, if we have high humidity, so lots of water molecules around the leaf area, then that's not a very strong concentration gradient. And this means that the water molecules are going to diffuse slightly slower off the leaf. So if we have lots of water molecules in the atmosphere around the plant, that means we have high humidity, and that means that transpiration rate will decrease, it will slow down. Now lastly, we have air movement. So we call this air movement because it can apply to anything in the natural environment. We're typically going to be talking about wind here. So as you increase wind or as you increase air movement, you're going to increase the rate of transpiration. And the reason for this is because if you're increasing more disruption around the leaf area, that means that you're going to increase the rate in which the water molecules can move from the boundary layer of the leaf. So if we increase air movement, that means we're going to increase the rate of transpiration. However, this is one of the few situations in which the plant cannot control. So this is one of the examples where a plant can lose too much water. And this is because the plant has no ability to control the rate of the air movement. So if air movement is happening too quickly around the plant, then that means that water is going to be leaving the plant too quickly, and the plant can't control this. So there is risk of the plant losing too much water here in very windy environments, and that could lead to a point in which the plant is incompatible with life. So all we're looking for you to do here is giving you an opportunity to have a little bit of creativity at home, really. So the factors that can affect the rate of transpiration is something that is very key, and we need to become very comfortable with discussing and even better explaining. So if you want to have a go at producing a poster, or more specifically a learning resource, so I want to give you a bit of freedom here and an opportunity for your creativity to run a bit wild here. If you can produce me some form of learning resource, so you could even potentially get a real plant and somehow construct a way or construct something around that plant that explains maybe what transpiration is, and especially what those factors are and how the factors affect the rate of transpiration. So lastly, we have looked at what transpiration is. So it's the movement of water and mineral ions through the plant. It takes place in the xylem tissues. It takes place in one direction from the roots to the leaves. We have the transpiration stream, which is that constant negative pressure due to the evaporation of water from the leaves that is constantly drawing water from the soil to the roots, to the stems, to the leaves, into the atmosphere. And we're having that nice cycle due to that negative pressure produced. We have looked at the 4 factors that affect the rate of transpiration and how they affect and why they affect the rate of transpiration. And lastly, we need to look at a practical. So in an ideal world, this would be set up on my lab desk, and this would be demonstrated to you, and we would be able to see, and you would be able to see this in action. However, we don't have that opportunity at the moment. So you will see this at some point over the next couple of years, but in the meantime, it's important that you get comfortable with the idea of how we measure the rate of transpiration. And how we do that is with a device called the potometer. So you can see from the diagram here, the potometer is essentially a collection of glass tubes that you have a beaker containing water on one end, and you have a capillary tube that leads up to a plant on the other. So with this, what we're going to do is we're going to give the opportunity for the plant to transpirate, or to transpire, sorry. So plant is going to be transpiring, and as it does that, its water is going to be evaporating from the leaves, and it's going to be needing to draw more water from somewhere. And the way it's going to do that is it's going to draw some water from the reservoir below in the glass tubing. So as that water moves through, what we're going to do is if you carefully tilt, so if you fill the tube up with water and then you carefully tilt the tube upwards, the right hand side up, you will introduce an air bubble into the tube. So what you want to do is very carefully, and there is a sort of clever little knack to this, but if you just introduce an air bubble into that capillary tube, and then what you can do is you can place a ruler along the capillary tube, and all you're going to do is then, as the plant draws water through the capillary tube, that air bubble is going to move towards the plant. So as more water is transpired, that air bubble is going to move from the right-hand side to the left-hand side towards the plant. And what you can do is you can set a control variable of time and say, we'll do this experiment for 5 minutes. And what you can do is every minute you can measure how far the air bubble has moved. And then what you can do is you can then introduce a independent variable. So what we could do is we could increase the temperature. So we could have this experiment over 5 minutes at room temperature, around 24 degrees centigrade. And then what we could do is increase the temperature, so put it in an environment of, let's say, 30 or 34 degrees centigrade. And then do the same experiment again, exactly the same protocol. The only thing that's changed is the temperature. And then we can see how transpiration is affected with temperature. You could do the same with light intensity, so you could put a lamp closer to the plant, and then as the light intensity increases, so should the rate of transpiration, and again, the air bubble should move quicker than without the lamp being there. So this is a very nice and relatively simple way to measure the rate of transpiration, and I appreciate that in an ideal world you would be seeing this in a classroom setting. So what I have done is I've included 2 links below just to show exactly how the potometer works, and a demonstration pretty much exactly how I would be doing it in the classroom. So have a look at those videos just to get a good idea exactly how the potometer works, because you could always get a question in your exam that presents you with a potometer, and what we don't want to happen is you encounter that and not have a clue what a potometer is or how it works. So have a look at those videos and gather a decent understanding of how the potometer works, and then how we can measure the rate of transpiration.